This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
So we are all increasingly living in a virtual world, especially our kids. They're on Facebook, social media. People go on eHarmony, they compare their qualities, and they then meet and fall in love, etc. So why not do the same thing in immunogenetics? And so the virtual crossmatch is gaining a foothold because it does streamline the process and may accelerate the time to put in a kidney in patients. So our director of immunogenetics, Dr. Raj Raja, will be presenting the case for and the benefits and maybe the potential problems of the virtual crossmatch.
I'm on. Thanks, Flavio. Open my slides. So this is the virtual crossmatch, but this is the reality of most of the crossmatches that we do in the laboratory. So everyone in this room knows about the crossmatch. We have been doing this for years and years. It's very simple laboratory test, just taking the donor cells and mixing with the recipient and see any antibodies present in the recipient serum binds to the donor cells. So the readout will be positive crossmatch or negative crossmatch. Positive crossmatch, don't transplant, and the negative crossmatch, do the transplants. Very simple technique. But in over 50 years, people identify this very precise antibody identifications. What are the antibodies present in the serums and what are the antigens expressed on the donor? If you know this precisely, you can do it like a match cam virtually. We can do it virtual cross-match. So that's what I'm going to talk. It's going to be very simple, but to get into that level, see, the virtual cross-match slide, I have just only one slide. Rest of the 49 slides are building the story. So I'm going to talk more about basic immunology. The system that you have, everyone has it. And you are using it for every day, every time, on fighting against infections. So these are the objectives of my talk. In the end of my talk, I hope you will understand the basic of histocompatibility systems and various HLA antibody testing methods, various cross-match techniques, and ultimately what's the concept and utility of virtual cross-match. So if your recipient receives an allograft without any immunosuppression, they will reject. So the system, the genes, proteins, the factors that involves in this rejection process, we call it histocompatibility systems. It has three major components. We know this. So the blood group systems, which is very simple ABO group. Then the major histocompatibility systems, which are HLA molecules. And the last one is the minor histocompatibility system, which is any amino acid difference between two different individuals. But you can characterize all these three systems into two factors. So all three are polymorphics, very different between individuals. And they are immunogenic. They can trigger immune response. But the strength, the amount of polymorphism and the strength of immunogenicity is variable. For example, the blood group antigens, there are very few variations, only 4 different blood group antigens. But it's very high immunogenic. You don't want to cross this ABO barrier. But the minor histocompatibility complex, as I mentioned, see any single amino acid difference between two individuals are considered to be the minor histocompatibility antigens. but most of them are not making any immune response. Less in terms of immune response, they are very, very low. Only around 20-25 distinct minor histocompatibility antigens have been characterized that is known to elicit immune response. But the major histocompatibility antigens that are HLA, that in terms of polymorphism, they are highly polymorphic as well as highly immunogenic. That's the big barrier that we are trying to overcome. Because these HLA molecules are the HLA molecules are the one present the foreign peptides, the viral peptides to our immune systems. So every single cells in our body express HLA class 1 molecules. So if you get infection, so I'm hitting the wrong button. Okay, so if you get the infection, the infectious materials or the viral proteins is loaded in the context of our self-HLA class 1, which is present ultimately to the subset of T lymphocytes. It's called CD8 positive T lymphocytes, which we have a majority of T cells in our bloodstream are this type. So once the T cell recognizes the class 1 molecules with this foreign peptides, it triggers the immune response. Basically, it releases all the cytolytic molecules that kills these infected target cells. So you contain the infection. So if the infection is not contained by this mechanism, so then a subset of new cells come into place. These cells are called professional antigen-presenting cells, so like dendritic cells, monocytes, macrophage, and B cells, and so on. Their primary function is just capturing these all antigens and presenting to our another subset of immune cells. So to perform this special antigen-presenting functions, they express a special HLA molecules. They call class 2 molecules. So class 2 molecules are expressed on a subset of immune cells, antigen-presenting cells. But the class 1 is expressed on almost every single cell in our body systems. So once the protein is, the foreign peptide is presented in the context of class 2 molecules, it is presented in another subset of T cells called T helper cells, which basically secretes interleukin-2, which are the basically growth factors that can act upon the T cells and the B cells. So it can trigger more cells which are specific to this invading pathogen that is mounted on the class 1, class 2 molecule. So you can trigger immune response, you can contain the immune response. So if you, in the whole slide, so the two important, just one important factor, that is HLA molecules, HLA class 1, class 2, those are the one control your immune response. If you don't have HLA molecules, you will die by infection. So now we are living with many, many viruses and bacteria. So to present all this variety of pathogens, just one HLA molecule is not good enough. That's why we have many HLA molecules. We have at least 15 HLA molecules, we have it. And those genes are present in this particular chromosome, chromosome 6. And the gene complex is known major histocompatibility complex. This is the most gene-dense region ever identified in humans. It spans around 4 megabases of DNA. It contains 124 genes, and 50% of them are expressed, and 50 genes have some immune function, not necessarily HLA molecules, but it also triggers other proteins, like interferon gamma, complement factors, and all involved in the immune functions. And physically, this HLA or MSC region can be divided into 3 regions. Towards the one telomeric region is the class 1 regions. Towards the centromeric region is class 2 region. And in between these two are the class 3 regions. So in the next slides, I'm going to make it in a very simple picture of the same slide. So these are the class 1 regions, and class 2 region, and class 3 regions. So in class 1 regions, we have 3 genes, HLA A, B, and C. And class 2 region, we have 3 sets of genes, DR gene, and DQ gene, and DP gene. So the DR genes, in each in the set, we have one gene, A gene, and other one is B gene. So the A gene is the one encoding alpha chain, and the B gene encodes beta gene. These together combined and expressed in one DR molecule. So you have one DR, one DQ, and one DP coming from alpha chain and beta chain. And all these genes are polymorphic. So the number, except one gene, the DRA genes, which has only two variants. And the number I put underneath each gene indicates the number of different variants identified in population. So the HLA-B is very highly polymorphic, over 2,459 different HLA-B molecules identified in human populations. So you will have either one or two out of these 2,500 different varieties of HLA-B molecules. So now we have two chromosomes, one from father and one from mother. So we look at the gene content, it's not always same. And particularly in the DR regions, you have additional genes. Some haplotypes having additional DRB genes. Either it could be DRB3 or DRB4 or DRB5. So it is even the gene content can be different because out of 4 haplotypes, you will have 2 of any of these 2 combinations. So in terms of genes, one can differentiate. Then what I'm going to show you here is this is 2 different individuals. The individual one here in the bottom having simple genotype. There is only one DR gene, but the individual on the top having 4 different DRB genes. So what happens if you consider if all the genes are polymorphics, then you will have in the bottom ones, the B locus, you have the same allelic polymorphism, B27, CW7, everything same. So in this case, you will express only one B molecules, one C, one A, and the combination here from the alpha chain, beta chain express one DR, one DQ, and one DP. But if you look at on the top, the second individuals, you have polymorphism in every single genes. So in the B locus, you have B7 and B44, and similarly, every C locus, you have 2 different alleles. So in this case, you will express 2 different B molecules, 2C and 2A. Okay, so that's a huge difference. One individual having only 3 HLA class 1 molecules, other individual can have 6 different HLA molecules. And if you look at the DR, the combination of these 2 genes can produce 1 protein. Combination of these 2 genes can produce another protein. And you have here and here 1 protein, another one. So there is a 4 possible DR one can express versus just 1 DR by 1 person. There will be a huge diversity. Some individual can express only 2 proteins, some individual 3 proteins, and so on. And if you look at the DQ, both genes are polymorphic, not like the DR, both genes are polymorphic. So that the combination of these 2 one protein and this 2 one protein and the trans, the combination of the top gene, the DQB1 here, and this gene can make 1 protein, another one. So you will have another 4 DQ molecules. Similarly, the DP, A and B are polymorphics, then you will end up with expressing 4 different proteins. So just looking at the number, one individual can express 18 different HLA molecules, another individual can express just only 6 HLA molecules. And this is the diversity in HLA systems. This kind of diversity, extensive diversity can be seen within the families. One child can have only 6 HLA molecules, other one can have 18 different HLA molecules. So what it tells, going back to my original slide, the biology of HLA is presenting antigens. So that indicates clearly this individual having more HLA are advantages, having presenting a variety of pathogenic peptides to the immune system when you compare with the individuals having a limited HLA phenotypes. But the problem is in transplantation, finding an HLA matched transplantation is very hard. So what I've shown here is different varieties of HLA matching algorithms. So the top one, see here in this combination, so these are the recipient HLA types. So for simplicity, I just show A, B, and DR. So you have 2 A's, 2 B, and 2 DRs. The donor type is same, so we call them HLA identical. But if you look at here in the bottom, the recipient donor types are entirely different. So the organ has one HLA type and the recipient has different HLA type. So if the organ gets infected, so there is no biology of antigen presentations. So there is different varieties of, there can be one mismatch or haploidentical and so on. So I'm not going to talk about these mismatches, but the better mismatches are the better outcomes. So the worst mismatches are the worst outcome. So in transplantation scenarios, if the kidney expresses this set of HLA molecules, if the recipient also have the same HLA, there's not much problem, no alloreactivity. But there is a difference. For example, this molecule, the HLA-B27, is different in the donor. Then all the immune cells will be targeting here. So the T cells, CD8 T cells, will recognize and trigger cytolytic activity. And the B cells will recognize this difference and triggers antibody production. And the CD4 T cells, if there is an additional class 2 mismatches, it recognizes and triggers growth factors for the T cells as well as the B cells. So you will enhance all the immune responses against all the mismatched HLA antigens. So now still the transplantation can be done, because if you remove all these cells, the immune cells, you can put your kidney. So that's what we are doing. So the induction therapy that basically eliminates all the immune cells. But still the problem is the antibodies. There's no drugs. All these drugs are resistant to the antibodies or antibody-producing plasma cells, which is hidden in the bone marrow. So we end up with having problem with these antibodies. Antibodies are very bad because it is basically the people having transplantation produce antibodies. The people having multiple pregnancies are producing antibodies, and the transfusions, infection, vaccination also can trigger HLA antibodies. And the antibodies can react in many different ways. It can fix the complement, kill the organs. It is kind of adapted that connects both innate and adaptive immunity. And it has a lot of consequences. It can goes from the hyperacute rejection to the chronic rejections. And it can also eliminate transplantation. If the patient is highly sensitized, it is very hard to find a suitable donor. So to Paul Terasaki, in 50 years back, he developed a very simple test. That's the Crossmatch. Still, people are using it in the laboratory. It is just mixing the lymphocytes from the donor and serum from the recipient and see whether any antibodies in the recipient is binding to the donor cells. So if there is no antibodies in the serum, recipient serum, so nothing binding to the donor cells, if there is an HLA antibody that binds, then he adds the complement that kills the cells, so that if you add the dye, so you can distinguish the dead cells and the live cells. So the dead cells means you have the antibody in the serum against a particular HLA molecules. So you don't want to transplant here. And here the live cells means there's no antibodies, so one can do the transplantations. So this is very simple method that he developed in 1960s. Then he also published his paper. So he looked at 30 positive cross matches. 24 out of 30 are showing hyperacute and accelerated rejection. And most of the negative cross-match individuals, the graft function was fine. So since this publication, it becomes the standard of care for the transplantation. But the problem here is these two numbers. So, but still, the cross-match is used as the clinical testing, but it's not too sensitive or not too specific. There are some false positive and false negative reactions. So people try to eliminate this false positive and false negative reaction by simply modifying the test procedures. They can eliminate. So if you look at the cells are not only expressing HLA molecules. They express other junk of proteins. And the same way, the recipient serum not only having HLA antibodies, but it also have other antibodies, including IgM antibodies. So the IgM antibodies can kill the cells. It can, because it can fix the complement, but it is not clinically relevant. So people using this chemical, the DTT, one can deplete all these IgM antibodies, or they can add anti-human globulin to enhance the sensitivity of the cross-match testing. But still, it is not good enough. Still, there is a lot of false positive and false reactive reactions. So then people develop the flow cytometry cross-match, which is 30% more sensitive than the classical CDC cross-matches. So it's the same technique. So you have the donor cells, then you add the serum mix with them. If the serum has HLA antibodies that binds to the donor-specific cells, then you add, instead of complement, you add the secondary antibody that is specific to HLA, the IgG antibodies. The secondary antibodies has a fluorochrome, which can be detected by the flow cytometry. So the more antibodies it binds, the stain will be high. So you can detect in the flow cytometry, and that goes, the reading will be read by like this. Then when you compare with the negative control serum, which do not have any antibodies, then detecting this negative control serum picked with this positive or the patient's serum, you can come up with a number, how much the median channel is shifting for the T cells and B cells. So that gives a value of a channel shifts, a quantitative readout for the positive cross matches. So it's not going to tell just positive and negative, but you can also see in terms of number how much positive. Then because the secondary antibodies are very specific to the IgG molecules, they don't recognize IgM molecules. It's very, very specific to the IgG molecules. And also adding the pronase, one can deplete all the background. So you get very specific cross matches. But still, there are a lot of false positive and false negative cross matches. So 88% of the flow cross matches are false positives. So that means you will avoid unnecessary transplantation unnecessarily. Then 7% of the flow cross matches are false negative. That means if you have the DSA, it's not going to detect in the cross match. So still we need to have a very refined cross matches. So that's what the virtual cross match come into play. So there are two things needs to be done for the virtual cross match. If you know what are the antigens expressed on the donor sites, if you define all clearly, everything, all 12 different HLA genes, some of them are, sometimes you may need to define at the allelic levels. Then in recipients, if you characterize all the antibodies using single antigen bead assay, which I'm come to later. So if you define these components from the donor side and recipient side, you can predict what are the antibodies. You have it in the patients, and is that antibodies are against the donor's antigens. And how much antibodies, if you know that number, you can predict how pretty much whether it's going to cause the CDC crossmatch or the flow crossmatch, or it's going to be a negative crossmatch. So there are many methods we have been using it, define the HLA antigens and antibodies. This is the Luminex technology. This is the mainstay of our laboratory. We have been using for last 10 years. So this technology uses polystyrene microbeads. Then we don't do this anything. We just buy it from the commercial companies. So basically, they color them using two different fluorescents, yellow and red fluorescent, with a different proportions, so that individual beads, these 100 beads, can have a unique colors. So the flow cytometry can distinguish those beads. Then you take the beads and conjugate with this unique HLA protein. So each bead has a specific HLA molecules. Then we pull all these 100 beads, which has 100 different HLA molecules into a single tubes. So this is what we buy from the commercial products. Then once we get the patient serum, you isolate the serum and mix with these beads, the same way that we do for the cross matches. Then if the patient serum has any antibodies, for in this case A2, that binds to this particular bead. Then you add the secondary antibody, which has a fluorochrome. Then you can detect using the Luminex methods, which has two lasers. One detects the population of the cell base or the beads. Other one detects the secondary antibodies. So again, it's a very simple test, but it's very specific test. So if you run the negative control serums, it looks like this. So you have different 100 beads. Each bead is coded with a different unique HLA proteins. Then in the x-axis, it gives the fluorochrome, the MF5 values. That gives how much antibody it binds. So if you look at this is the patient serum showing some antibodies here. There are 6 or 8 different antibodies you can see. It's over 20,000 MF5. And if you look at all these antibodies, you can say there are different antibodies, but in reality, there's only one antibody here. So there are 5 different antibodies, And the frequency of these antibodies, or the target for these antigens, are 62%. So that means if the patient having this profile will avoid 62% of the UNOS donors. And they are basically one antibody. I'm going to explain in a couple of slides next. So if you look at all the HLA molecules, they are very, very similar. So the differences are very, very, very little. So each HLA molecule has their own unique epitope. So unique epitope is nothing but the one recognized by the antibodies. So each HLA molecule here has a unique epitopes which are recognized by HLA antibodies. Then in addition to this unique epitope, the HLA also has a public epitopes. For example, here, this triangle is present in all these 5 HLA molecules. So if an antibody is developed against these triangles, that particular antibody can react with all these HLA molecules, okay? So for example, if a person getting a kidney, so this is HLA type, the allograft HLA type, and the recipient HLA type, the mismatch antigen is only one antigen, that is HLA-A2, then we'll think he will develop only A2 antibodies, but actually he's developing more antibodies because of the shared epitopes. So the antibodies, it's not like one HLA molecule and one HLA antibodies. You have one HLA molecules and multiple HLA antibodies, and the same antibodies can bind to different HLA molecules. So we call them as a cross-reactive group. So there are 11 different cross-reactive groups has been identified. So it's always, when you have the patient's antibodies, never been just single antibodies. You have a bunch of antibodies. And some of these cross-reactive groups are very, the frequency is very high. So it is present in multiple or majority of the HLA type, particularly the BW4 and BW6. So they are present in 1/3 of the HLA B molecules. For example, this patient, so you can see 1/3 of all the HLA class 1 molecules are reacting. So basically, this is only one HLA antibody, so that is BW4 antibody. So the antibody binds to that particular epitope shared by all these HLA proteins. So the calculated PRA would be 61%. So if the patient, this kind of antibodies, he need to avoid 61% of the UNOS donors. So the BW6 is another epitope, very broadly present epitopes, which avoids 80% of the donors in the UNet. Some individuals can have 2 epitopes or 2 cross-reactive groups or multiple cross-reactive groups. So these are some unique cases. So for example, if the woman having BW4 genotype, and her husband was BW6, then by multiple pregnancies, she will develop antibodies against this particular epitope, BW6. Then that means she will develop many antibodies, then she will avoid 85% of the UNOS donors. So again, this is one single antibodies, but binding to multiple HLA class 1 molecules. So the problem is most of the time we cannot even detect these antibodies. So you can see all this clumping into this one corner, but the MF5 values are less than 1,000, very weak MF5s. So most of the time we don't even call these antibodies, but they are in the bloodstream, so it can trigger the memory response. So the cells that the plasma cells that making antibodies are in the body. So we don't even call this antibody, but now we are realizing we should call this even the weak antibodies less than 1,000 MF5. But some individuals can have antibodies to almost everything except their own molecules. So what I'm showing here is just only the class 1 antibodies. If you see the class 2 antibodies, so majority or 30% of the waitlist population having or sensitized, having some level of antibodies. So in addition to that, there are some allele-specific antibodies. You have one A2 molecules, but not there are 200 different varieties of allelic polymorphism of A2. So the people can develop antibodies to a particular HLA A2 types. So you need to distinguish those antibodies also. That's another level of complexity. So if you look at our wait list, so So over 11% of the individuals in the kidney wait list having 80% over 80%. It's a huge problem. It's not the high PRA is not very random. It's over 600 people having 80% PRA, and most of them are retransplant or the woman. So what is the outcome of all this antibody testing? So once we have the antibody testings, we will list all the unacceptable antigens in the UNet. You can just click all this button. If you have, say, A2 antibodies with the patients, you click them, that will avoid any UNOS donor having HLA-A2 antigens. So that's what you can do it in the laboratories. So for example, if the patient has a A2, so these are the donor types here. So if they have A2, so all you have to look at how many individuals carrying the HLA-A2 molecules. So in this panel, there are 47% of this panel carrying HLA-A2 targets. So you need to avoid all this 47% of the individuals. If the patient has additional antibodies like DR4, then you will be avoiding more donors. So that's going to be 60% of the donors. So if he has additional antibodies like a DQ5, you'll be avoiding more donors. That's going to be 69% of the donors. So the more antibodies the patient carrying, so you will avoid a lot of UNOS donors. So getting a matched donor is very unlikely. So we also did a correlation between the MFI, the strength of antibodies, and the cross matches. So this is the flow cross match, T cell cross match, and the MFIs. So if anything over 5,000 MFI, the flow cross match will be positive. And anything over 15,000 MFI, the CDC Crossmatch will be positive. So in UCSF, we block anything over 2,000 MFIs. So that means you never get any positive crossmatch, even with this sensitive flow cytometry. But the problem is there are some allele-specific antibodies, and there are some I think I have this slide. So if the patient has this unique antibodies like a DP, DP alpha, DQ alphas, and allele-specific antibodies, the UNOS has no option to block it. So we cannot block those antigens, so we will be getting the positive cross-match. And on the top, we are always looking at the antibodies in the patients and donor typing in the donors. We never look at the expression of the donor types. So the physical cross-match is the only way that is donor-specific. So that's still, it is a very valid cross-match because the HLA proteins, the expression can be different by donor to donors. It is not very well characterized, but at least some locus, such as like HLA-CW, is known to be low expressed alleles when you compare with other B and A molecules. And some alleles like a B44, the expression can be very low when you compare with other HLA-B molecules. So it can be an allele-specific expression also. In addition to that, other cytokines can induce the class 1 and class 2 expression. So it is important to have the donor-specific cross-matches done. And moreover, in the solid phase assay that we are using, the single antigen-based assay, it has its own problems because all this single antigen has been synthesized using, majority of them are using insect cell lines. So it is not comparable to the native proteins. So the antibody can bind to the conformational epitopes like here. So when the synthetic proteins are processed and conjugated on the beads, this conformational epitope can change. So that's why you need to have the physical cross-match also done. So that will be more appropriate. So this is the protocol that we used in UCSF. So you see, if the patient has no antibodies, we don't enter any awards in the UNet. So it can be done virtual cross-match. If the patient has antibodies, particularly with these unstable antibodies, we screen multiple times, it goes up and down, then we want to put in these categories. Then if the patient have the DP antibodies, DQ alpha, DQ beta, or allele-specific antibodies, too many antibodies, then we will avoid only those with over 2,000 MFI, and we will follow with do the flow cytometry cross-match. And if the patient antibodies are well-defined and are stable, then we will avoid all antibodies. Anything over 1,000 MFI, then we'll go for the virtual cross-match. So this is not only the practice in UCSF, but many other centers also using the virtual cross-match and very successful. So these are the advantages. It adds precision to the actual cross-match, and it eliminates the physical cross-match. So that means you save a lot of money and workload from the laboratory as well as the OPO. And there's no samples involved, so you don't need to wait for the samples from the donor, and you don't need to do the cross-match in the middle of the night. Then it improves allocations, and it increases the rate of transplantation, particularly in the sensitized people. And it permits a national paradigm change, and also one can count the risk of memory response. If you know what is the memory mismatches from the previous transplant, you can avoid those antigens also. So in comparison to this, the virtual cross-match is very powerful technique. So if you define the antibodies and antigens in a precise manner, you can predict your cross-match very well. So I think I'll stop here, then I'll take any questions. Thank you. Yes, Dr. Rossi.
I had a question of why the beads actually ever work. Because the polymorphism between HLA molecules really resides in the peptide binding group. And the differences between one allele versus another really affect the motifs of the peptide that's being bound. And you referred to it really quickly, that these single antigen beads are made in insect cells.
Most of them.
Most. So the issue is it because we're so conserved compared to insects that most of the cell peptides, because in your picture, you didn't draw the peptide in, you just drew empty class 1 and class 2. But there are human-specific peptides and even tissue-specific peptides that may be in there that you're not picking up with the single antigen beads. So I can see why there might be a disconnect. It seems like a small number, but there could be a disconnect completely between the single antigen antibody testing and an actual cross-match where you're using actual cells that actually really truly express that stable trimolecular complex between either alpha chain, heavy chain, beta-2 microglobulin, and the antigenic peptide. Can you address why it actually works? Are most of the peptides exactly the same between the insect cells or whatever peptide being loaded on?
No. As you indicated, is that the peptide doesn't affect the antibody binding. It is the most of the epitopes recognized by the antibodies are in the peptide binding group as well as outside the binding groups. It's not deeply buried. So there are some polymorphism that is recognized by the T cell epitopes as well as around the antigen binding group. So most of the antibodies are hitting those regions. And some the public epitope, the BW4 and BW6, they are deeply buried in the inside the pocket, but it's down, it's in the corner, in the C terminal end of the pocket. So still the antibody can access. If you look at the antibody, so they are the complement, or not complements, the loops are the one recognizing those antibodies. So there are two, there's a publication test, they take two HLA molecules, B molecule, B44O2 and B44O3. They are naturally occurring alleles. They differ by a single amino acids, but their polymorphism sitting in the binding group. But you can see the difference in the single antigen binding. So it's still, yes, but when you make the antibodies, still the biology is not clear. But so far there's a correlation, that's it. So there are a lot of differences in terms of the antibody binding. So that's why people still argue the physical cross-match is necessary. I mean, it's not very studied. Like the DP antibody, you can see 20,000 MFI, it never binds to the cells. But sometimes at 2,000 MFI, it gives a positive cross-match.
If a recipient has a pre-existing immune complex disease, like lupus, with a very highly reactive anti-DNA, how would that complicate the cross-match?
So they probably have the more auto antibodies, so not necessarily directed to the HLA molecules. So they are clinically insignificant, but it can affect our testings. So what we do, we clean up all the cells using the pronase that chop down all the FC receptors that non-specifically bind antibodies. So that clean up a lot of false positive crossmatch. We do see the B cell crossmatch is positive. When you do the pronase, it becomes negative. Okay, thank you very much.